Good morning, Avisek.
Hi, good morning.
So, good morning, Dr. Sharaj Ghosh. Shall we start? Okay. Okay. So, good morning, everyone. Good morning for our speaker, Dr. Avisek Banerjee, and good evening to everyone. Today, we have assembled in this evening for one more precious seminar. It's again a pleasure to have you all around for this interactive session being organized by the Zoological Association of Burdwan. Today we have Dr. Avisek Banerjee, an expert in molecular biology, currently associated as a research associate in the Yale School of Medicine, Yale University, United States. Dr. Banerjee pursued his master's in zoology from the University of Calcutta, and then he completed his doctoral degree from the Bose Institute, Kolkata. He has associated with Tata Translational Cancer Research Center as a postdoctoral fellow. Briefly, he was the recipient of several fellowship, international travel grants, and attended numerous conferences and delivered talks. Dr. Banerjee has authored and co-authored numerous publications in peer-reviewed journals, which are of international repute. Let's give him a big hand and begin with the session. Please, Dr. Banerjee.
Thank you very much for your generous introduction. Let's present my screen. Okay. Oh no, just a minute. I'm having a trouble in sharing my screen. Something is not working. Can't share. I'm sorry, I have to leave and have to log in with another computer because this is probably not working. Sorry for that. I will be back in like 2-3 minutes. Okay. Sorry for that.
Okay, please go ahead. Dear students, I think the speaker is having some problems with sharing the screen. So please hold on. He will be back on time. On behalf of the Zoological Association of Burdwan, I'd like to re-announce that our speaker was having trouble in sharing the screen. So I think we got our speaker back, so he will be starting soon. Thank you.
I apologize. My apologies for the delay. And so how, I have this PowerPoint open on my desktop, but
Did you try, Avisek, did you try with sharing the entire screen? Like you have some options, right? Your
just a minute.
entire
So
screen instead of a window.
screen.
Yes, please.
Yeah. So I'm sharing my entire screen. You can see my screen now. Just
Now it's coming, I think. But there's a mirroring, yeah.
Okay, can you see?
Yes, we do, yeah.
You see my PowerPoint?
Yes, we do.
Okay, thank you very much. Okay, so at any point of time, if you have any problem in viewing my presentation, please let me know. So first of all, thank you very much to all the organizers of the Zoological Association of Burdwan for inviting me, particularly Dr. Shamdas Bandyopadhyay and Dr. Manavesh Majumdar. And I really look forward and appreciate their initiative in organizing this online lecture series for the undergraduate studies in zoology. And I've seen the list of speakers, and they're very impressive. And I really don't know if I fit in that category, but I feel privileged for coming here and sharing with you a topic which is very close to my heart. So I'm going to talk about the diversity of animal life with an emphasis on phylum Porifera. Now, I worked on, after passing out from zoology, as Dr. Bandyopadhyay said, I actually got interested in doing research in cancer biology. And I did my PhD studying epigenetic regulation of genes in cancer. And presently at Yale also, I'm focused on doing research in cancer, but from a cell biology point of view. So why did I choose a topic when I was invited to share my thoughts with you? I choose because I felt I got a chance to recapitulate my old subject, which was so close, and still it is so close to my heart. And so by using this platform, I would like to reach you, try to ignite your interest in studying classification, because classification, I believe, is the best knowledge we can have as a zoologist, as a student of zoology. We have this added advantage compared to other fields of biology. We know classification, and this is very important. I mean, to me at least, the scientific understanding and the inspiration that I have developed later in my research years have came from the basics of classical zoology. So without further ado, let's start. So first of all, what's the need of classifying animal kingdom? Now, we have a lot of animals around us. So classification will definitely facilitate the identification of organisms. And this is very important. For example, you go to any trekking, you go to any forest, you see a new animal. You're going to get afraid of it, or you don't know whether it's a friend or foe. But if you have an identification, oh, this is a good animal, it won't be very harmful to me, right? So identification of organisms is very important. Second is it helps to establish the relationship among various group of organisms. Suppose we want to conserve an animal, right? But we don't know whether that animal is going to survive alone in nature, or it needs some other group of animals to help them survive and sustain. So for a sustainable biodiversity, we have to we have to have the knowledge of the relationship among various group of organisms. And that's how classification of animal kingdom is very important. And we as a zoologist, I repeat, have the privilege to know it, to study it. The next is helps to study the phylogeny and evolutionary history of organisms. See, we are all interested to know the history. I mean, anything, like we need to know how we look like 50 years back. What happened to Burdwan University 50 years back? So in the same way, we are interested to ask questions how animals have evolved. And from a big picture mind, why do we need to know that? We need to know that because if we know how animals have evolved, we know what are the forces have worked behind their evolution. And so identifying those players will help to understand whether in this present scenario, are these animals going to go to extinct or these animals are happy? So again, from the conservation point of view, how the animal have evolved, what are the forces of evolution have worked? If that knowledge is with us, then we can have the power to conserve us nature, okay, our animals around us. And the beauty of this animal classification is that by studying a few animals, the characteristics of the whole group can be known. So you don't have to go into the field and study each and every individual. If I say, okay, this animal, I got a new animal from a field study, and I can identify, okay, this is a vertebrate, so I will go deep into the characteristic, into vertebrate characteristics, not thinking of any other else. So these are the need of animal classification. So the next question is how animals are classified. Now, if you have any questions in between, please feel free to unmute yourself and ask questions at any point of time, okay? So, and I'm going to talk about now is how animals are classified. Now, it's very simple. First, animals can be grouped based on their similar features. And then after grouping, these features can be differentiated whether they're primitive or modern. Now, if they're primitive, that means they have likely evolved earlier in the history, and modern means they are evolved more later. And they are also supported by fossil records and genetic analysis. Let me give you an example. You're very familiar with this kind of phylogenetic tree, right? Say A, B, C are three groups of animals. Forget about whether they're phylum, subphylum, species. Don't think about it. Say A, B, C, and three groups of animals, okay? And B and C are they're very close. That means they have some similar features. That's why they're grouped together. From the evolutionary point of view, we think that they have or they share a most common ancestor. Okay, this is purely an imaginary thing. Don't get confused that what is this ancestor? Which is this ancestor? Why don't I see this? Don't think about that. Just for example, I live in USA and I see two Indian walking in the street. Oh, I think these are from India. If I see two Europeans, so these are from Europe. So just like very crude basis, they have been grouped together, okay? So now the B and C have some similar features, which are called, they have some different terms, which I'm not going into details. And compared to A, so A also have features which are not as similar as B and C, so they are grouped separately. And so A also have a common ancestor, but probably that have evolved more early than B and C's common ancestor. Now, how do we make it more simple? You turn this phylogenetic tree like this. You have this root and the bottom, okay, root of the phylogenetic tree. You pull it down, okay, you just pull this down, imaginary, and how do you see the change? You see the shape, it changes like this. Now you can add another dimension to this, the time scale. So as I said, animals are first grouped together, and that is supported by fossil records. So these animals, B and C, are grouped together, and probably A has evolved earlier in the time scale, which is also supported by fossil records. And then we can say, okay, A is ancestors to B and C having some primitive features. And B and C are likely they are more modern animals having modern and present features. Now, when I say having a primitive and modern features, what do I mean by that? Let me give you some examples. It will be very simple. See, any feature, say take about an example like a body symmetry. See this image. There are three kinds of organisms. These animals, they don't have any symmetry. Don't think about the examples right now. Just look at the picture and you will very clearly understand that they don't have any symmetry. That means There is no particular line that can divide this animal into two equal halves, which are mirror image of one another. Okay, that's called a line of symmetry. In these animals, they are called radial symmetry because they can be divided into several equal hubs passing through the center axis of the body. Okay, and these animals, they have bilateral symmetry. That means they can be divided only through one single plane. Now, to a zoologist, this is very easy. If you ask which is a primitive and which is a modern character, obviously the bilateral symmetry is a more modern character. Why? Because having a bilateral symmetry gives a directional movement, a better movement towards a food or to run away from the predator. Okay, so this is a more advanced or modern feature. So just you have this concept. So I will be talking about Porifera. So if you have this kind of concepts, it will be easier for you to understand classification, appreciate the science behind it. And the ultimate goal is to remember it, memorize it. So if you do this logically, so I'm just trying to give you a little more examples. Think about body cavity. We know body cavity or coelom, right? So this group of animals, they don't have coelom. This group of animals, they have true coelom. That means the coelom is in between or split of the mesoderm, in between the mesodermal layers. And these animals, they have coelom, but it is between mesoderm and endoderm. We call that pseudocoelom. So again, to a zoologist, if you ask which is a primitive and modern characteristics, obviously having a true coelom is a modern character. Because coelom houses several organs. It can act as a hydrostatic skeleton, whatever it is. So again, if you take another example, the fate of blastopore and development of coelom. So in some group of animals, the blastopore, which is actually formed during the developmental stage, with development, it forms the mouth. But in another group of animals called deuterostomes, same blastopore forms the anus first, and opposite side gives rise to mouth. So again, if you look into this animal, the classification, you can clearly understand that protostomes are primitive and deuterostomes are modern. And so, for example, the body segmentation. Some animals, like today we're going to talk about Porifera, they don't have any segmentation at all. But some more advanced animals have segmentations, like arthropoda, they have tagmata, and even humans. And so this way, if the lists actually go on, so you can take basically any characteristics like locomotion, habitat, mode of development, alimentary canal, and you can ask yourselves whether this is a primitive or modern characters. And that's the basis how animals can be grouped together. And once they're grouped together, you get this phylogenetic tree. And so this is very simple because today we are going to talk only about sponges. And so this group of animals is very primitive compared to the rest of the group. But if you have a logic behind studying this animal diversity, it will be very easy for you. At least to me, it was very easy. When I actually understood the logic, I got really interested to pursue and memorize this whole classification. So the animals which are very primitive, they don't have, they are just only diploblastic and then becomes triploblastic later, and other characteristics. So let's talk about phylum Porifera. So these are the very primitive organisms, which are mostly marine. They're obviously all are aquatic and mostly marine. So if you have any questions so far, just feel free to unmute yourself and ask. Okay. So the general characteristics of phylum Porifera, these are very, very simple organisms. The body shape is variable. There's no specific shape to it. They're mostly cylinder shaped. And the symmetry, also there is no specific symmetry. As you see, the next phylum, the Nidaria, they will have this diploblastic and in between have mesoglia. Then the symmetry becomes radial symmetry. So if you imagine how this evolution takes place, it will be very very good for you to memorize. So then the further general characteristics is the surface of the body has numerous pores called ostia. Now, this is one of the very classical feature of phylum Porifera, where the name actually comes from perforated. Body is perforated with numerous pores, or these are called ostia, for the entry of water. So the water enters through these numerous pores in the body, and water comes into a central structure called spongocyl. I'm talking about that later. And then the water goes out from just one single opening called osculum. Okay, so the body has numerous perforations called ostia by which water enters, and just one opening, large opening, which is called osculum, by which the water goes out. The body layers, they are diploblastic, though it's not very well differentiated. And the cilium, which is the cavity, body cavity, is absent. So as we know that cilium, the body cavity, is, we consider cilium if it is only formed between the mesoderm. If it is formed mesoderm, this is called the true cilium. Otherwise, if it is between the mesoderm, endoderm, in some other phylum, it's called pseudocilium. So here the cilium is completely absent. But what is present is a spongocyl, the space in between in the body's cavity. Okay, so I will be talking that later. But what is the function of this? So as water enters through this ostia, the food particles also enters through this pore, and the water and the space of the spongocyl is lined with choanocytes. Okay, these are flagellated cells. Now the beating of the flagella creates a water current. So the water enters through the pore, and there's a water current created, which actually propels the water out from the osculum. Now in between that, these flagella also trap the food particles. Once the food particles are trapped, these food particles are carried inside the cells for digestion. Okay, so this is a very important feature of all poriferans. They have this numerous pores through which the water enter. Water is then taken out through osculum by beating of flagella, but flagella also traps the food, which helps in digestion. Now, the another feature of this sponges is the canal system. So this is called the canal system, which I said. The water, this is the direction of the water flow. So the water comes from outside, goes inside through this pores called ostia, and it reaches the spongocyl. But this is not a cilium. Okay, this is just a space inside the body cavity. It is not formed between the mesodermal layers. So this is not a cilium. This is spongocyl. These are very primitive organisms. And this spongocyl, and the water goes out through osculum. Such type of canal system is called asconoid. Now we have two more, okay? And again, if you know these types of canal system, if you know the different feature of sponges, it will be easier for you to do the classification and remember it, okay? So the other type of canal system is the cycanoids canal system, where the body is actually folded. And what does that mean? You can see it very clearly that this spongocyl is no longer lined with choanocytes or flagellated cells. The flagellated cells or choanocytes are actually restricted in this small chambers, which are called the radial canal. So this is a very big difference. And so the water, how does the water flow? Again, the water comes in through ostia, and this are called the incurrent canal. And by prosopile, they comes into this radial canals, and these spaces are called apopiles. So these are prosopiles and these are apopiles. Just have to remember some names, and then the water comes into the spongocyl and it goes out. Some group of animals have even more complex canal system. As you can see, this flagellated chambers do not directly connect to the central spongocyl. Okay, again I repeat, here the flagellated chambers are directly connected to this spongocyl, but here this flagellated chambers are connected to another flagellated chamber, and that is in turn connected to the spongocyl. So this is a more complex structure. And so the direction of water, as you can see here, the water comes through ostia, through incurrent channel, then to prosopiles, and then these are the flagellated chambers. But from here, from apophyles, they go to excurrent canal, and then this finally, these are the spongocyl, and then exterior. So just keep this, three types of canal system in mind. Then we'll come to classification next. The other important feature of these animals is their skeleton structure. Now, these are again very primitive organisms. They don't have skeletons or bones, but what they have is these are called spicules. Now, the spicules in sponges, they could be, structurally they could be different shapes and size. Mostly the shapes are different, and by the composition, they could be either made up of calcium carbonate or silica or protein. Okay, so based on that, they could be calcareous spicules. That means these spicules are made up of calcium carbonate, or siliceous spicules made up of silica, or spongine fibers, which is basically a protein. So again, based on their size, based on the shapes of the spicules and the compositions of spicules, this phylum has been classified. So just keep in mind, we will discuss that later again. So this is the phylum classification of phylum Porifera. It is divided into three classes: class Calcarea, Hexactinidida, and Demospongy. Now, as you can see here, I think you are now familiar with this type of cladogram. This is the root, and if you see, this is the kind of a timeline, not only supported by physical observation like features, but also supported by fossil records, as well as genetic analysis, which is kind of ongoing with the advancement of this modern molecular biology tools and techniques. And so, as you can see here, this during evolution, so the calcium carbonated, the poriferans having calcium carbonated spicules became Calcarea. Then later the siliceous spicules developed. Now the siliceous spicules, if they are 6-rayed, it becomes hexactinidida. So hexa means 6. So hexactinidida, spicules with 6 rays. And demosponges, these are also siliceous spicules or spongine fibers, but they're never 6-rayed. Okay, so this is the very broadly, there are three classes. Very simple: Calcarea, Hexactinidida, and Demospongy. So the first one is Calcarea. Again, these are exclusively marine, and they can live solitary or colonial, and the body is cylindrical. So for a classical example is Cycon or Scypha. Okay, so the cardinal feature, the main feature of this is that they are made up of calcareous spicules. That means the spicules are made up of, the composition of the spicules are calcium carbonate. Okay, they have two orders, Homocoela and Heterocoela. Now Homocoela, that means they are calcareous poriferans which having asconoid sponges, asconoid type of canal system. The other Heterocoela, they have cycanoid or leuconoid. Now if I go back, so this is the asconoid type of canal system, and the other two are the cycanoid and leuconoid. So here, the Homocoela, they have these asconoid sponges. So the other two features automatically follows it because it's very simple. The body wall is thin and not folded internally, and the body cavity or spongocyl is lined with collar cells as schwannocytes. So these are actually the characteristics of asconoid sponges or asconoid type of canal system. And so it's easy to remember this Heterocoela is cycanoid or leuconoid types of sponges. That means the body wall is thick and it is folded internally, and that's why the spongocyl is not lined by choanocytes. The choanocytes are actually restricted in those flagellated structures, if you remember from the picture. The spongocyl is lined by only epithelial cells. Okay, so just to remember and just have some examples also. The next class is Hexactinidida. Again, from the name, it's very easy to remember that these are having 6-rayed spicules, but remember, these are not calcareous anymore. These are siliceous spicules. They have two orders. One is
Hexastereophora, and the other one is Amphidiscophora. Again, this class is very easy to remember. If you see the name Amphidiscophora, that means how their spicules look like. Based on that, this order has been named. So if you see the picture here, Amphidiscophora means the amphidisc. They have this two 6-red spicules attached to the end of a long stick. Okay, so these are amphidisc spicules. Amphi means on both sides. But in case of the other order, Hexastereophora, they never contain amphidisc spicules. So all the spicules are 6-red. You can see here the 6-red spicules, but they don't have this amphidisc type of spicules. So that's the very cardinal feature of these two orders of Hexactinellida class. Okay, so just remember a few examples. Any example in each of these orders. The last but not the least is Demospongy, and this is a big class. So the Demospongy, the body is again very irregular shape, asymmetrical. The interesting thing is that the spongocyl is absent, and the skeleton, the exoskeleton, is made up of siliceous spicules or spongin fibers, or both, and interestingly, or none. They sometimes don't even have that. So if you ask me that, okay, they don't have spicules, then how can that be Porifera? I mean, they have other features of Porifera, like their body is perforated by numerous pores or ostia. So I'm not mentioning that here because all the class have features which are already present, in the phylum. So the next is another subclass is Tetractinellida. So the Tetractinellida, as you can see, the name, the tetra means 4. So these are the type of poriferans which have tetraaxon type siliceous spicules. Okay, the tetraaxon is 4-red. Okay, so this is the tetraaxon spicules. And the other one is they have this leuconoid type of canal system, the most complex type of canal system. Okay. The next class is Monoaxonida. Monoaxonida means they have monoaxon spicules. So these are very, very interesting. With the name of the subclass, you can actually find the characteristics, and that's how it can be easy for you to remember. Tetractinellida means 4. Monoaxonida means just 1 monoaxon spicules. And the third subclass is Keratosa. They don't contain siliceous spicules. They just have only spongin fibers. So going back and to Tetractinellida, they have actually 3 orders. And so as you go down the classification for any phylum, initially it becomes very easy because the features are very cardinal features and easy to remember. As you go deep into the classification, the different features become very subtle, very small. The features very overlapping. I mean, to a very amateur person, it's overlapping. But if you asked a very specialized person who have spent years of research in Porifera, probably these are not overlapping. These are very, very separate and distinct features. Anyways, at least when I was a student like you, it was very difficult for me to understand and remember. But I'll try to be as clear as possible. So the subclass Tetractinellida, they have 3 orders: Microspongida, Carnosa, and Choristida. So the first order, they do not contain endoskeleton or spicules. As I said, the spongin fibers can have spicules or may not have spicules as well. The other order, they contain spicules, but they are very small in size. But the last one has spicules which are very much developed into megascleres and microscleres. So these are, the ways of differentiating the 3 orders in this subclass of Tetractinellida. The other subclass is the Monoaxonida. So Monoaxonida means they have these siliceous spicules which are single rayed. Okay, this is just a stick. They have 4 orders, and the first order is the Hadromerida. This is very easy to remember because they have the broad end is knobbed. So you can see here, there's a knob-like structure at the end of the spicules. The other Helichondrida, then Poecilosclerida and Haplosterida, they have also different types of spicules, where in some, like microscleries, are absent. Here the microsclery spicules are present. So these are some of the features. And finally, the Keratosa, which I already mentioned before, that this group of poriferans, they don't have siliceous spicules, but they have a network of sponging fibers. So I think with that, I talked about how animals are classified, what's the importance of animal classification. I mean, these are very, very basic. I mean, there is a huge lot of informations, but I just wanted to, imbibe some interest in you. So if you just read them, if you think about the big picture, why are we reading animal classification? What's the importance of it? How it can contribute, how this knowledge can contribute to my understanding, then I think it becomes more interesting to study and definitely to remember. And then I covered some of the general characteristics of phylum Porifera, and then I classified phylum. I also talked about the classification of phylum Porifera. These are some of the books which I find very interesting and very informative to look into. And I think that's it. Thank you for your kind attention. If you have any questions, feel free. Anything.
Students are requested to ask questions.
Sir, should I ask?
Yes,
Sure.
please go ahead, Saptarshi.
Sir, from the first day of our course, we are talking about spicules forming endoskeleton of sponges, but I have not found any source talking about how they are forming the endoskeleton of the sponges. Can you say something about it?
Okay. So basically, so as I said, like spicules are basically the endoskeleton. So the term endoskeleton is important. Endo means inside. So definitely, if you look into this animal kingdom, there are two types of skeleton. One is endoskeleton, which is inside the body layers, which cannot be seen from outside. And the exoskeleton is, for like in arthropoda, there actually the skeleton is outside, and the muscles are attached inside of that skeleton. And so these are the spicules. So spicules are, if you see any poriferan, if you take example of this, Euplectella, the spicules are actually present inside and these are like microscopic structures. And so these images have all scale bars. Scale bars means they are in some micron size. So if you dissect these animals, see it on the microscope, I think you can see these spicules. And so these are the spicules. These are present, and these give the supportive structure to this animal. If that answers your question.
Sir, are they interlocking or they are attached to some kind of fiber or proteins?
Well, that's an interesting question. I really don't know, and definitely they are not floating. There has to be, if they are supportive structures, then so these are very oversimplified images we find in the book chapters. So your question is whether they're interlocking and they're supporting structure, and I think I guess so. They are. They are just like any other skeleton structures which give support to the body. I think they should be interlocking structures which give support. But again, I am not very much confirmed with that.
Okay, sir. Thank you.
If you have more questions, you can ask.
Sir, I have a question.
Yeah.
Sir, what are the possible ancestors to sponges?
So this is again, if you look into the phylogenetic tree, at the top of my head, I don't remember it right now, but definitely could be some animals which are diploplastics or having a body layer not very well differentiated. Definitely those animals have to be aquatic. Definitely those animals have to be very primitive in terms of, say, they don't have body cavity and all that thing. So if I see a transition from Porifera to Nidaria having, in between the ectoderm and endoderm, there's a mesoglia. They're from locomotion power, they have locomotion power, the evolution of locomotion power. The evolution to have a different sex, sex is a different, not sex is a different, they can actually have a sexual and sexual mode of life cycle. So if I imagine the animals from which poriferans have developed, they would likely be, diploblastic, or their body layers would probably not be well differentiated. They should be aquatic. But again, there could be some information out, but I don't know, honestly, what could be the animals.
Okay, sir. Thank you, sir.
I think if there is no more questions, so we could express our thanks, and I think that's a nice presentation, Dr. Avisek Banerjee. So Dr. Saroj Ghosh will express the vote of thanks. Dr. Ghosh, please go ahead.
We are delightful to you, sir, for your attractive, commendable lecture and spell out your valuable perception with us. I think your esteem talked on Porifera and how our animal classified will be adequate for our students in their academic endeavor. We extend our heartfelt thanks to you for accept our invitation and spend your valued time with us. Also, thanks to my dear students for attending the lecture. Thank you, everybody. With these few words, I conclude the session. Thank you.
Thank you very much.
Thank you.
Thank you, sir.